Hey everyone, today we're going to get started talking about oxidative phosphorylation. So here we will focus on kind of the final step of energy production and the actual production of ATP in animals. So we already looked at carbohydrates, we looked at lipids, and we looked at amino acids and how they can be reduced um, for fuel in the cell, right? So the actual molecules themselves are oxidized and then they reduce electron carriers such as NADH or FADH2, which carry electrons that then can be utilized to make ATP. And they're utilized to make ATP via a whole complex called oxidative phosphorylation. Here's a very large overview diagram of the idea of cellular respiration in full. You can see energy is flowing from our initial energy molecules, such as, again, amino acids, fatty acids, sugars like glucose. These go through their representative paths. Sugars and glucose go through glycolysis, fatty acids break down in fatty or beta oxidation. Amino acids will be broken down via a numerous amount of paths, one of them being the urea cycle. Uh, and most of these are going to lead in eventually to our molecule acetyl-CoA, which then goes through stage 2, which we've talked about, uh, oxidation of acetyl-CoA via the citric acid cycle. This produces even more energy electron carriers, NADH, FADH, and these then enter stage 3, which is the transfer of electrons and final oxidative phosphorylation to produce ATP. And you can see again as a large overview, this is the flow of all the electrons coming from various areas. Again, these electrons are just representative, but remember that they are being carried by molecules like NADH and FADH2. And in animals and in cells for respiration, the way that we generally are going to convert this electron energy into ATP is through this respiration, the electron transport chain, in which you will note you're going to use oxygen and protons. Now, this isn't hydrogen gas, just the actual protons themselves, so acidic protons, to make water. So one of our byproducts to make energy is water. And we have to use oxygen. That's why we need to breathe in oxygen. Again, one of the other byproducts that is expelled through this whole process, again, to remember, is at numerous steps carbon dioxide. So the actual formation of carbon dioxide is not directly connected to stage 3, but the intake of oxygen, the need for oxygen, is related to stage 3. Now, this process of oxidative phosphorylation is broken up into two components that are connected. And we're going to be talking about each of these. The whole of this process, again, is called oxidative phosphorylation, but we're going to look at each of the stages. The first being the actual electron transport chain, which is, as it sounds, a chain of proteins in which electrons are transferred through them, where they will get to their final destination. And then the second portion, which is chemiosmosis, where you're actually making the ATP. So note that the electron transport chain, its use is to make an electrochemical gradient, a difference in electron potential between a membrane. And chemiosmosis is then going to use that gradient you created by transferring electrons to then make ATP using a special protein. The actual making of ATP, taking ADP and adding a inorganic phosphate, is of course highly thermodynamically unfavorable. It's the reverse of a very favorable step, right? Breaking ATP releases a lot of energy. It's very favorable. Going backwards is very unfavorable. So how do we achieve this? How do we make ATP if it's very thermodynamically unfavorable? Phosphorylation of ADP is not a result of the direct reaction between ADP and some high energy phosphate carrier. So the energy needed to phosphorylate the ATP again is going to be provided from the flow of protons, that's H pluses, down an electrochemical gradient. Again, we're going to use the electron transport chain to make this gradient. That's where the energy is coming from. It's literally coming from the flow of an electron gradient. Remember, just a reminder, a gradient occurs when you have a buildup of some molecule on one side. So let's say you have a lot of this molecule A, and we have a barrier here. There's only maybe one or two A's over here. We now have built a gradient in which there's a difference. And if you open up a channel here, naturally, through a chemical means, this will reach equilibrium, right? So you'll actually flow A into the other chamber. So our body is going to construct a gradient so that we can use the flow, almost like the power of a waterfall, that flow of the gradient to power making ATP. The proton gradient that is needed for ATP synthesis is established across a membrane that is impermeable to ions. It has to be, because we do not want them to just be flowing through at random and causing equilibrium to occur. We want them to build up and then gate them, as I just showed, so that there's only a small area for them to flow, and we can utilize that small gated area to create energy. In different organisms, the membrane that is used is different, but the idea is the same. This idea of creating this gradient across an impermeable membrane, something that's impermeable to ions. In bacteria, for example, it actually happened in the plasma membrane. For animals, it's going to occur in the mitochondria, specifically in the inner membrane of the mitochondria. And in plants, where this is being utilized for photosynthesis, it actually happens in the thylakoid membrane, and that's part of the chloroplast. Chloroplasts are essentially the mitochondria, but for plants. These membranes must contain proteins that couple the downhill flow of the electrons with the uphill flow of protons. So again, we're going to be dumping off electrons, but we need to move protons to make a gradient. You're going to see this when we in detail look at the electron transport chain. The idea is, as the electrons get dumped off, we're going to move protons across our barrier, making a gradient. And finally, in all of these cases across species, there has to be some type of protein that then takes the protons that we just made into a gradient and couples it to actually making the ATP. So at this point, we need some proton that utilizes that flow, this waterfall effect of our protons moving back through the gradient. We need something that captures that energy and uses it to make ATP, almost like a mill. If you think of a water mill that utilizes the energy of flowing water, we have a protein that does the same thing, utilize that flowing energy to then make ATP. Okay, so this is the theory. It is technically a theory, although we do have a lot of detail about how the electron transport chain works. There are still some small areas that are we're unsure of exactly how things are happening. But the idea is our mitochondria as seen. Here is our inner mitochondria membrane. And then there's, of course, the outer mitochondria. So this is actually happening again in an inner portion of the mitochondria. The idea is that we are going to dump off the electrons from our donors, NADH and FADH. These are going to flow through some series of proteins. These are right here, these proteins here, these 4 proteins. And as the electrons flow through them, the electron transport chain, they pump H plus from one side of the membrane to the other side. And you actually get all of these H pluses, this gradient on one side. So you keep doing that, and you pump a lot of protons onto the outside of that inner mitochondrial matrix or membrane. This process also is going to use oxygen. As we said, we have to use oxygen to make water during the electron transport chain. Then the natural flow of these protons back to the inside, because now we made a gradient, is going to occur through our channel protein, which, like a mill, is going to use the energy of that flow to then generate ATP from ADP. So that's the overview of the process. I want to go over some of the details of what is going to happen in this whole process. The actual mitochondria is important to understand because its structure is how we achieve this gradient. There is an outer membrane of the mitochondria itself, which is porous to many metabolites. It's relatively porous. It allows metabolites to go in and out. We've talked about transfer of things through the mitochondria. For example, fatty acids do need to go through a special protein to get into the mitochondria. Some metabolites can just freely flow in. For example, we have our malate aspartate shuttle, but some cannot freely flow out or in. And thus you need to kind of transform metabolites to enter or leave. But generally it's relatively porous. Things can get in and out either via some just free flow or via some specialized protein. there is the intermembrane space, or the IMS, of the actual mitochondria. It's kind of like the cytosol of the cell, but now we're even closer into the cell. So again, you have your outer membrane. You have this inner space in here. This space is going to be where we have high proton concentration, because ultimately it's where we're going to build up H plus. So it thus has a low pH. It's a very acidic area. There is then the inner membrane, which we just showed. You can think of it inside the mitochondria like this. This one is very impermeable. Ions cannot just freely transfer through it. And it is the location of the electron transport chain complexes. So as we showed in this previous picture, they live actually on the membrane itself. This is not just a nice, perfect circle of an inner membrane. If you ever look at pictures of mitochondria, you usually see these kind of squiggly lines drawn in them. That's because the inner membrane is very dynamic looking. It's not actually just one circular oval on the inside of the mitochondria. It has these bends in it so that it actually increases the surface area. That means there's more membrane within the inside of the mitochondria. There is the actual matrix itself as well, which is where the citric acid cycle, parts of lipid and amino acid metabolism also occur. So we've talked about those areas. Again, this is going to be inside of that inner membrane, and this is where we have low proton concentration. So there's not that much H plus. So there is actually a higher pH. It's a little bit more basic. All right, so here's kind of a close-up look of what I just went over. You can see an opening of the mitochondria, the organelle itself. Here's the outer, right? This is the outer membrane that we're showing. And then on the inside, we have this inner membrane. And you can see, as I said, it's not just a perfect oval, squiggles around a lot. And this is, again, to increase that surface area. The outer membrane, again, pretty permeable. Inner membrane, very impermeable. You need transport molecules to get things into it. Again, we talked about the pH, the difference of what is occurring. Things that are going to happen as far as breakdown pathways are occurring mostly in the inner matrix that we talked about. So we talked about conversion of pyruvate into acetyl-CoA, pyruvate dehydrogenase complex. That's in the matrix. Things for the citric acid cycle, all of those enzymes, that's inside the matrix. Fatty acid beta oxidation enzymes, so breakdown of fatty acid, that's happening inside the matrix. Amino acid oxidation happening inside the matrix. There's also some DNA. Mitochondria has its own DNA, which we will talk about later in this semester. Ribosomes, and there are many other enzymes that live here too. So the inner matrix is where most of the action is actually occurring. And the reason that we want to have all of that inside the inner matrix and have this electron transport chain on that membrane, we want them nearby so that we can quickly transform all of those electron carriers that we make from these processes into ATP, right? They don't have to move very far to go from being made in the citric acid cycle to then going to the electron transport chain, dumping off their electrons, and then eventually making ATP. All right, so let's look closer at the actual electron transport chain. The electron transport chain is made up of a series of enzymes, large complex proteins, and they are electron carriers. They transport electrons. Each of these complexes are going to contain multiple centers for reductive oxidation reactions, so redox centers. Some of them actually have bound molecules that are flavin molecules, so parts of the molecules that even make up FAD2 and NADH, which again, these are carrying the electrons free floating. Portions of them are actually also bound as cofactors into the enzymes within the electron transport chain. So don't get confused about that. We have our free carrying FADH2s, our NADHs, but we also sometimes have these flavin cofactors bound into the electron transport chain. The electron transport chain also uses cytochrome molecules. We've seen cytochrome molecules before. Remember, these are inorganic molecules that are utilized as other types of carriers. For example, heme is a type of cytochrome, but they're a general molecule that is used as a cofactor in a lot of enzymes. Another one is an iron sulfur cluster. So these are three of the major types of centers, kind of redox centers, in which our electrons will kind of transfer from one to the other as they go through this chain. So here's a close-up of some of the carriers we'll be talking about. Just a reminder that FADH2 and NADH2, they both can carry 2 electrons. The flavin portions of the molecules, which can be covalently bound into proteins, again, they can act to funnel and to distribute electrons as the electrons move through this chain. They can accept 2 electrons from a carrier and donate electrons one at a time. This idea is very important. Again, we're going to talk about movement of electrons, how many electrons are in each step, how those electrons pump protons. So I want you to remember these ideas of how the order of how electrons can be transferred, how many electrons can be transferred. So for these types of FAD molecules, they can only transfer them one at a time. They can't do 2 at a time. Cytochromes, which also are utilized as well. Remember, these tend to be iron containing. In this case, iron containing porphyrin rings. There's also cytochromes A, B, or C, which really have the same base structure as you can see here, but just differ in the types of additions that are on their kind of side in the blue. So you can see these are the portions that are different, but the main ring structure is the same. These can only carry 1 electron at a time. And here's our iron sulfur clusters, a view of them. These also can only carry 1 electron. They are coordinated in the enzymes themselves by cysteines. Cysteines are where the sulfur comes from in the iron sulfur cluster. And of course, they then coordinate actual iron molecules to make this clustered region. Generally, it's going to be an equal number of irons to sulfur atoms, and there are different types of iron sulfur clusters. You can see even just as simple as with 1 iron, 2 irons, or up to 4 irons with multiple sulfurs coordinate them. And again, the sulfurs aren't just free-floating sulfurs, they're portions of the protein where a cysteine is that is then using its sulfur to hold that iron in place. This is kind of a view of what a protein would look like. You can see here is that coordinated cluster of irons. Now, another carrier we will be talking about a lot in the electron transport chain is called coenzyme Q or ubiquinone. This is what ubiquinone looks like. It's actually a molecule that has a generally polar head group, and that is where it can carry electrons. But it also has this very long lipid tail. This is important because this duality lets it travel through areas that are very lipophobic. And I want to remind you that most of this is taking place on the membrane of the mitochondria. What are membranes made up of? They're made up of lipids, right? Don't forget that when we talk about cell membranes, any type of membrane, they're made up of those phospholipid groups, right? Where they have some usually phosphocholine, phosphoinositol, different types of phospho head groups that have lipid tails, right? And the membranes are just a sandwich of these. You have head groups on each side and this lipid center. So ubiquinone, coenzyme Q, this molecule, this lipid portion of it, this lipid tail along with the tail, lets it move inside the lipid membrane, which you will see is how it actually moves its electrons around. So this makes it unique in this sense because other types of carriers that we just looked at can't actually move on the inside of the membrane where it's all very lipophobic. Coenzyme Q can also carry a total of 2 electrons. Again, this is oxidized one at a time, but ultimately it can carry 2 electrons. Again, the main portion I want to point out here is that it can freely diffuse in the membrane. So that means it can carry electrons and protons with it. It picks up 2 electrons, but it also picks up 2 protons, as you can see here. It's an electron and a proton. And by doing this, carrying those electrons and moving it through the membrane, it can diffuse electrons and also those protons, which don't forget, we're pumping protons in this process to the other side of the membrane. You'll see this is going to be kind of like the carrier between complexes. Often coenzyme Q is going to connect each of the complexes that's part of the electron transport chain, moving electrons from complex to complex, kind of like the shuttle. So here's an outline of each of the complexes that make up the electron transport chain. There are 4 in total, and they are not all always utilized, and we'll go through when that is the case. The first complex, complex number 1, is called NADH dehydrogenase. And it is where NADH drops off its electrons. You can see it uses a flavonoid type group and an iron sulfur cluster. The second one is succinate dehydrogenase, which we have already seen. And I'll remind you, succinate dehydrogenase also is part of the citric acid cycle. Back in the citric acid cycle, I mentioned that one of the complexes in the citric acid cycle was unique in that it was actually bound to the inner mitochondrial matrix, and that is where it was held. it has a dual job of helping in the citric acid cycle and being part of the electron transport chain. It uses a bound flavonoid and an iron sulfur cluster. The third group, the third of the electron transport chain, is called ubiquinone or cytochrome C oxoreductase. It's often referred to as just cytochrome C. It is probably one of the most well-known complexes within the electron transport chain. And it utilizes heme and also an iron sulfur cluster. Remember that heme is just a type of those special cytochrome molecules. So it is a cytochrome. The last of the electron transport chain complexes is cytochrome oxidase, similar name. It also uses heme, and it is a bit special in that it uses copper, some copper centers, copper A and B centers, which we'll see when we get to that. Okay, so the first complex, NADH ubiquinone oxoreductase, this is one of the largest macromolecule complexes in general in mammalian cells. It has about 40 polypeptide chains. So remember, it's actually made up of separate proteins that are then interacting together using this quaternary structure as a whole complex. The way that it is encoded is very interesting. So the genetic information for this NADH complex, complex 1, is actually in both your nuclear DNA, the DNA inside the nucleus, but also in parts of the mitochondrial DNA, which, as I mentioned slightly, mitochondria actually has its own DNA. It's bound into the matrix side, so the inner portion of the mitochondria. As I noted, it uses a bound flavin. It also uses iron sulfur cluster, and it can accept 2 electrons from an NADH. This is where NADH will drop off its electrons to then enter into this electron transport chain. Those iron sulfur clusters, as I mentioned, can only pass electrons one at a time through this complex. Here's what it looks like and an overview of what is happening in complex 1. Again, complex 1 accepts electrons only from NADH. So it is the drop-off point for NADH. NADH is going to go back to its oxidized state of NAD+. As it drops off its 2 electrons. Remember, NADH carries 2 electrons. These 2 electrons are then held on by this flavin mononucleotide complex. Remember, it can hold 2 electrons. They're then passed through a series of iron sulfur clusters, one at a time, all the way through to the end of the complex, where they are then given to a ubiquinone, or again, our coenzyme Q. It's usually represented by this Q. Notice we call the unbound version Q, and the bound version is called QH2. That's just a reminder that it picks up 2 electrons and 2 protons. So here's 2 protons that needed to get picked up by our coenzyme. Now, during this whole process, the powering of this electron movement, which is happening in this arm of the complex, powers the movement of protons from the inner matrix to the outer inner membrane space of the mitochondria. And it powers a total of 4 protons. The how of how this happens, how this matrix arm and the movement of electrons is connected to powering those protons is still unclear. But we do know that electrons move through this portion, and somehow that powers this then movement of protons. So in whole, what is happening? Again, we're dropping off an NADH, and it is going to drop off its 2 electrons to make NAD+, right? Ultimately, those 2 electrons are shuttled all the way to a coenzyme Q, where they are picked up to make a QH2, a bound coenzyme Q, which also holds on to 2 protons. Through that entire process, don't forget that we also are going to be transferring 4 protons from the inside to the outside. So here we are designating the inner matrix as N, just to be clear what we mean here, to that outer inner membrane space, the outer space portion of the mitochondria as P. So notice that though we take up these multiple electrons, so there's 1, 2, 3, 4, 5, 6 total that actually come from this side, the N side. Only 4 in the end of just this step end up on the P side, the intermembrane space. 2 of them are moved out, right? They're moved out of the inner membrane or the matrix side, but they're the 2 that are bound to that coenzyme Q. All right, complex 2. This is succinate dehydrogenase, and again, we have seen this complex in its other form before, talking about citric acid cycle. Succinate dehydrogenase is actually the acceptor for FADH2. So complex 1 is where NADH will drop off its electrons. Complex 2 is where FADH drops off its electrons. These 2 act separate of each other. Electrons are not going from complex 1 to complex 2. Electrons will go from complex 1 all the way to complex 3. Electrons dropped off by FADH will enter at complex 2 and go to complex 3. Now, these are coming from FADH2, where FAD will accept 2 electrons to it. Now, remember, there is another portion of this in which it is actually taking succinate and making fumarate. This is the portion of it that's connected to citric acid cycle. So some part of it is actually taking electrons from succinate and moving them onto an FAD that is bound to the complex and making fumarate. So again, I just want to remind you, it is a single enzyme, but it has dual roles. It has the role to take succinate and turn it into fumarate for the citric acid cycle. But it also has the role of capturing and donating electrons in part of the electron transport chain. So here is a close-up of complex 2, and we'll look at what is happening. It has this succinate binding site. This is where it is acting as the citric acid cycle enzyme. And here's that bound FAD that is part of the complex. It can take succinate into the binding site where it is transferring electrons to make fumarate, using these electrons then to shuttle through some iron sulfur complexes, eventually to a coenzyme Q or a ubiquinone. So again, these electrons are transferred from our succinate through the complex onto ultimately a ubiquinone. Again, our cytochrome Q carrier that is going to then enter into that inner membrane space. So this whole complex is going to ultimately use 2 electrons from that QH2 and reduce 2 molecules of cytochrome C. It's going to clear electrons from the reduced quinones via something called the Q cycle. Excuse me, this is for complex 3, what we just talked about, complex 2. So complex 3 is going to be taking electrons from those coenzyme Qs. Those coenzyme Qs, again, I want to remember you, came from either complex 1 or complex 2. The coenzyme Qs are then going to, again, act on a cytochrome C and additionally some iron sulfur clusters to then move more protons. Ultimately, they're going to move 4 more protons into the inner membrane space. So complex 3, here it is, it's very large. It again is going to be taking in some coenzyme Q molecules. Those are coming from complex 1 or complex 2, wherever those electrons were dropped off at. It is going to be using those coenzyme Qs, dropping off electrons, and ultimately it's going to move them to a cytochrome C molecule. This is the main takeaway. You're going to move it to a cytochrome C molecule, and through this process, transfer 4 protons through into the inner membrane space. Here's our cytochrome C, and I want to remind you again, cytochrome C only can carry 1 electron at a time. So for 1 incoming coenzyme Q, or QH2, for 1 incoming QH2, we have to then use 2 cytochrome Cs because there are 2 electrons on a coenzyme Q. Each cytochrome C only can move 1 electron. This whole process, I simplified it very much into just you're moving electrons onto a cytochrome C. This whole process is actually called the Q cycle, and it's made up of many more steps. Again, ultimately, 4 protons are transported across the membrane per the 2 electrons that you actually are going to get from the coenzyme Q and that reach a cytochrome C. 2 of those 4 protons that are going across the space actually come from the coenzyme Q. I want to remind you that there are 2 protons riding along with that coenzyme Q when it is bound to electrons. So 2 of those that ultimately get transferred across came from the coenzyme Q that is coming in. this cycle is going to be the model of where those additional protons are kind of coming from and picked up from the matrix. So I'll show you an overview of the whole cycle. Ultimately, what is happening is you have one incoming coenzyme Q. Here it is, right here. Here's our QH2. The QH2 enters into this complex 3, where it is going to systematically release electrons. You can see one electron is shuttled off onto this iron sulfur cluster and eventually to one cytochrome C. Remember, cytochrome C only carries one electron. The other electron actually is going to go deeper into the complex where it gets put onto an empty coenzyme Q. Coenzyme Q, remember, can hold up to 2 electrons. So here, this is a coenzyme Q, excuse me, and is just holding on to 1 electron. So it's holding on briefly to that 1 electron. The process of this first coenzyme Q coming in, dumping off its 2 electrons, each electron going down a different path, also is going to release 2 protons into the inner membrane space. Those 2 protons came from, again, our coenzyme Q. Now, that 1 electron that was kind of hanging out on our bottom coenzyme Q, it just kind of halted there. Now we have like a slightly radicalized coenzyme Q. We have to do something with that one. So what's going to happen in the second stage of the Q cycle is we now are going to use 2 protons coming from the inside and another incoming coenzyme Q. So here comes our second coenzyme Q. It's going to do the same thing. It's going to dump off its 2 electrons. 1 electron goes out and binds to a cytochrome C. And that cytochrome C can go off. The other electron goes down and it finds our radicalized coenzyme Q, the one that's already holding 1 electron from last time. This coenzyme Q can then pick up 2 protons. It now has its second electron and it makes a QH2, a full QH2, which goes back into the inner membrane. During this process, just like in the first stage, we release 2 protons in to the membrane, the inner membrane of the mitochondria. So this actually looks like you have to use 2 incoming coenzyme Qs. You ultimately are going to be releasing these 4 protons. You're taking in protons from the bottom, and ultimately 2 cytochrome Cs are moving. Now, the overall net reaction actually shows you only using 1 coenzyme Q, 1 QH2. 1 coenzyme Q ultimately giving up both of its electrons to 2 cytochrome Cs, then also taking in 2 protons from our matrix side and dumping out a total of 4 protons onto the inner membrane side in the overall. Why do we only sum this up as using 1 coenzyme Q? I'll show you because it does look like you have to go through these 2 stages where you put in a coenzyme Q and do it twice. which is true. You put in a coenzyme Q here, you get out an empty coenzyme Q here. And you put in an empty coenzyme Q here, you make a half radical coenzyme Q here, right? So here's an empty that came out, here's an empty that went in. So ultimately, we put in a full coenzyme Q, we get a half radicalized one. On the second stage, we put in a coenzyme Q, we take our half radicalized one, and we actually come out with a full coenzyme Q. We also come out with one empty coenzyme Q. So here our radical one was used up, and we actually also used up and made a full coenzyme Q in this step. So ultimately what this looks like in the net reaction is that we used a full coenzyme Q and we produced an empty coenzyme Q in the whole of the net reaction. So cytochrome C is now our carrier. After we end complex 3, we now have our electrons on a cytochrome C. This cytochrome C does not move inside the membrane like our coenzyme Q did. It instead is going to move through the intermembrane space. It's why it's actually shown on that side, on the intermembrane space side. And again, it only carries one single electron at a time. Cytochrome Cs are named actually based on their wavelength. And one way to tell if it is bound to an electron is via its absorption. So oxidized cytochrome C has a slightly different absorption than the reduced cytochrome C. And the position of this absorption band actually comes from this, or gives it its name. So if the position of the band is here, we call this alpha cytochrome C. If the position of the band is here, we call it beta cytochrome C. This is gamma, and this is the reduced form of cytochrome C. Now, complex 4, the last complex. This is where all of the electrons are now going to be shuttled. Remember, electrons can be dumped off from NADH onto complex 1. They can be dumped off from FADH to at complex 2. Those are both dumped off into coenzyme Qs, which go to complex 3. Complex 3 shuttles those electrons onto some coenzyme Q, or excuse me, some cytochrome Cs. And those cytochrome Cs are now going to finally end up at cytochrome oxidase, our last complex, complex 4. This complex, again, has some special units. It uses copper in it to do its job. So here's kind of a very overview of complex 4. You can see it has these copper centers that are actually held by some cysteines. It also utilizes heme on the inside. The overall, what's happening in our last complex is that 4 electrons are going to ultimately be used up. So it has to take in 4 cytochrome Cs, and those will reduce 1 oxygen molecule into 2 water molecules. Again, this is the net reaction. During that process, we also are pumping 4 protons from the matrix to the intermembrane side. So here is the overview of this complex. You can see we have to intake a total of 4 cytochrome Cs, each of those carrying 1 electron. Those electrons are then going to be transferred through these copper centers and the heme. So we have this kind of like iron copper center where we have heme with its iron, and the actual coppers that are bound in there in the center of the enzyme. That is where the actual oxygen is going to sit. Remember that heme is really good at carrying oxygen. So it makes sense that heme would be used as the portion of the enzyme that binds oxygen. So our oxygen is going to kind of bind into these hemes. And as our electrons are funneled through, the oxygen is then broken down to use water. Now, to make water out of oxygen, we need protons. So to be clear, 2 protons have to be moved in, but these are just used to make the water. Here's our 1 water. 2 protons are also pumped all the way through during the process to create our gradient. So ultimately, per water molecule that we make, we're going to be pumping 2 protons all the way through to the intermembrane space, and also moving 2 protons out of the matrix, but those are just used in making water. So again, per 1 water molecule that is made, we pump 2 protons out, and we also use 2 protons to make the water. Here is a full summary of that electron flow in the respiratory chain, just to see all of what is going on. Complex number 1, this is where NADH drops off its electrons. It is going to pump a total of 4 protons just in complex 1. Complex 2 doesn't pump any protons. It is the site where FADH can drop off its electrons. It also is part of the citric acid cycle and uses succinate to make fumarate. This makes a coenzyme Q, just like complex 1 does. These coenzyme Qs from either complex 1 or complex 2, eventually enter complex 3, where per a coenzyme Q, we are going to pump another 4 protons into the outer membrane or the intermembrane space. We also take 2 from down here during this process. Lastly, the cytochrome C carrier that is made from complex 3 goes to complex 4, where we generate water from oxygen, and during the process, pump 2 more protons into our intermembrane space. Here's the overall reaction of what occurs for each electron carrier that could enter the whole amount of protons that are pumped. If you drop off an NADH, I'm going to show you again, NADH enters at complex 1, where it pumps 4 electrons. It makes a coenzyme Q, which enters complex 3, where it pumps another 4 electrons, that's 8. Those cytochrome Cs, that's going to be 2 of them, all eventually enter to complex 4, where they will then pump another 2 protons. So that is going to be a total of 10 protons that is pumped, 10 protons that are pumped if you start with NADH. If you start with FADH, FADH only enters at complex 2. It doesn't enter complex 1. It doesn't pump those 4 protons at the beginning. It enters complex 2. It goes through 3 and 4. FADH thus only pumps a total of 6 protons. The difference in protons from entering at complex 1 with NADH versus entering at complex 2 with FADH is why NADH makes more ATP ultimately than FADH. It all comes down to the fact that NADH pumps more protons versus FADH. That is why we say NADH makes a total of about 2.5 ATP per NADH. FADH makes about 1.5 ATP per FADH2. Okay, we're going to finalize by looking at chemiosmosis. We just went through the totality of the electron transport chain, how the electrons are dropped off from either NADH or FADH, the carriers they go through to ultimately end up taking oxygen, making water, and pumping a bunch of protons into the inner membrane space to create a gradient. Remember, the whole second portion of this now is going to be using the gradient to make ATP. That is chemiosmosis. Now, I'm going to stop for a second and we're going to think quickly about how many electrons would enter complex 1 if you had the complete oxidation of a fat, stearic acid. This is a fat that has 14 carbons and 0 double bonds. So you have to think first about how many electron carriers are produced from a stearic acid and how these electron carriers can then enter into the electron transport chain. So this is asking how many would enter complex 1. That's a key factor here because what kind of molecule enters complex 1? Only NADH. So the first question is how many NADHs would get produced from this fatty acid? Well, we do know that in beta oxidation, we would make 7 acetyl-CoAs from this, and we would generate 6 NADH. We also would make 6 FADH as well, but we're not considering those for this question because we're only looking at NADH. So 6 NADH just during beta oxidation, but also 7 acetyl-CoA. Now, acetyl-CoA is going to also go through a process of being broken down through the citric acid cycle, and that is going to further generate some NADH. Remember that per acetyl-CoA, we are going to generate 3 NADH in the citric acid cycle. So that's going to be 21 NADH that get generated just from going through the citric acid cycle. So all of these NADHs are also, if you remember, carrying multiple electrons. Each NADH carries 2 electrons per NADH. So that means for these ones that came from the citric acid cycle, that is a total of 42 electrons. And these NADHs that came just from beta oxidation, that's a total of 12 electrons that would be carried by these NADHs. So for complex 1, how many electrons actually enter complex 1 from those NADHs? Well, it would be a total of the 42 from the NADHs we produced from the citric acid cycle and the 12 from the NADHs we produced from just beta oxidation. Which you can see that is going to be 54 electrons. Another question, thinking about the electron transport chain. So succinate dehydrogenase is dysfunctional in a species of slug, this type of garden slug. So its metabolism is compromised on a number of levels, but it can still undergo oxidative phosphorylation. The slugs can. What is the maximal ATP for these organisms if NADH is used as the electron source? Well, this question has noted that only succinate dehydrogenase is dysfunctional. Does NADH use succinate dehydrogenase? It does not utilize it when dropping off its electrons. So it's the ATP that can be made from an NADH is still exactly the same when thinking about the electron transport chain. So we still can utilize those 2.5 ATP for an NADH because NADH does not utilize complex 2, only FADH utilizes complex 2. So again, this is just that overview to remind you of the amount of protons that are pumped, each of the complexes, and what is happening at each complex. For chemiosmosis, all of these protons are going to cause a gradient to occur. As you can see, we've now pumped a bunch of protons with a positive charge to one side of the mitochondrial matrix, and thus have left a negative charge on the other side. This causes a electrical gradient, right? Positive on one side, negative on the other side. So we have a dual gradient that is going on. We actually have a chemical potential, and that's just because the pH itself is different, right? We have a acidic pH on the inner membrane space, but we have a basic pH on the inner membrane space, or the matrix side. And we have this electrical potential that we made, a positive charge on one side and a negative charge on the other side. This dual gradient is the driving force through our proton pump, which is then going to make ATP from ADP as we move our protons from one side through this gradient into the other side. Now let's talk a little bit about this gradient and some problems that can occur in making it. One of them is via reactive oxygen species. Reactive oxygen species are oxygens that are radicalized. They hold a radicalized electron on them, and they can interact specifically with the function of the mitochondria and damage this area the most. So, the reason that these reactive oxygen species are dangerous, they can occur in the mitochondria easily because that's where we have oxygen. And secondly, they can then damage our energy production by damaging the mitochondria. So, ubiquinone specifically, or coenzyme Q, is naturally what we call leaky, and it can facilitate the partial reduction of some targets of complex 3 that are not its normal targets, and ultimately results unintentionally in making free radicals. So oxygen, which is normally supposed to be bound in our complex 3 and made into water, can unintentionally react sometimes with coenzyme Q, ubiquinone. If ubiquinone moves out of that nice lipid space, it can then interact with oxygen. Now, this can then cause issues in damaging the cell. And this has to be dealt with via some molecules we have for processing oxidative stress. We've talked about these already. They are glutathione. The glutathione is used to correct um, the accidental free radical targets and the peroxide that is ultimately made from our free radicals. Glutathione takes peroxide and it makes it into water, correcting the damage. But glutathione itself needs to be processed. Once it takes on that electron and takes on the free radical, it goes from a reduced state to an oxidized state. And to keep doing its job, it needs to be turned back into its reduced state to go then help take some peroxide, make it back into water. So again, when we have oxidative stress, we are going to take it from its reduced state and make it into its oxidized state. And if you remember, the one molecule which is used to help in this process when we have oxidative stress is NADPH, which if you recall it's a product of the PPP, the pentose phosphate pathway. So there is a enzyme, nicotinamide nucleotide transhydrogenase, which allows for the transformation of NADH into NADPH. This is only on the inside of the mitochondria. I'm going to remind you that the pentose phosphate pathway, which is an important generator of NADPH, is actually in the cytosol. So when we have this damage occurring inside in the mitochondria, and we have to deal with it, we need NADPH there in the mitochondria. And one way to get it is by converting NADH into NADPH. So we're taking away from our energy reserves to deal with oxidative stress and take this peroxide and make it into water. Now we are going to further look at that actual proton motive force. So again, these protons are mostly being pumped from our complexes 1, 3, and 4. These, if targeted by inhibitors of the electron transport chain, of course, can ultimately disrupt oxidative phosphorylation and disrupt ATP or energy generation. There are a few known inhibitors that you're probably familiar with. One of them is cyanide. So cyanide can actually bind into the heme of our complex 4, stopping the ability to create, ultimately finish using that oxygen to make water and pump its final protons. There's also some inhibitors like antimycin A and rotenone, which those are going to target either complex 1 or complex 3. Now, if you inhibit the electron transport chain, you of course are going to ultimately affect ATP synthesis in some way. Just noted that all of the gradient that is made from the electron transport chain is what ultimately drives the production of ATP. So that means that ATP synthesis, actually making ATP, requires the electron transport chain, right? You have to make the gradient in order to then make ATP. But the electron transport chain does not, in reverse, require ATP synthesis. The electron transport chain can continue onward if you have accidentally uncoupled or in some way damaged the ability to make ATP. Electron transport chain does not need ATP to continue, right? All it does is take in electrons from carriers and move those electrons, and then through that process, pump protons. And it will continue to do that unless um, there is some problem with the actual enzymes in the electron transport chain, or if there is some stoppage of things like oxygen, which is needed for complex 4. So here is the actual complex ATP synthase that is responsible for taking that gradient and then making it into ATP. ATP synthase really does act kind of like a mill. In fact, it rotates, and through this rotation, it is moving ADP into becoming ATP. The rotation is driven by the movement of protons from the inner membrane space back into the matrix. So it actually has two subunits. One, the F1 subunit, is a complex inside the matrix that is actually catalyzing ATP. So that is this portion here. It sits actually inside the matrix, and that's where the ATP is generated. And the F0 or F0 portion actually sits in the membrane itself. It acts as the funnel, it acts as the gate, and it is going to drive the rotation of the enzyme. It is going to be the part where the protons move through and drive the rotation to make ATP. Here is the F1 portion. Again, this is the portion that actually is going to be making the ATP. You can see it has this hexamer shape that is actually kind of three portions, three dimers that are going to change their shape as they rotate and make ATP from ADP. There is one part of it that is always empty, the empty portion. So here's the dimer that's empty. There's one part of it that is bound to ADP. And then there's one part of it that has the ATP that has just been made. And essentially these just rotate around. So the ATP will be released and this portion will become empty. The empty portion will pick up an ADP and become the ADP bound portion. The ADP bound portion will rotate and make the ATP. So this just keeps going around and each of those sections go through the different phases. Again, this actually is occurring from a physical rotation that is happening as our protons move through the F0 subunit through that shaft portion and rotate the whole complex. The rotation is actually causing changes in the conformation of those enzymes. Pretty amazing that we're actually like physically moving the enzyme through rotation, and that physical movement is doing a chemical process. So you can see this is a kind of cartoon illustration of that rotation. Here is, this is some ADP. This is a subunit that has a finished made ATP. And this is the empty subunit. You can see one rotation, we move the rotation. And during this process, the ADP, here's our rotation here, the ADP becomes an ATP, right? Here is our ADP, here's the ATP. The ATP that was here is released, and this is now an empty section. And the section that was empty now is bound to a new ADP. This will rotate again, and the same thing's going to happen. Where our ADP was bound, we now have a bound ATP. Where our bound ATP was is now, it is released, and we have an empty section, and our empty section has picked up a new ADP. And this just keeps going around and around and around and making more and more ATP. Again, here's a cartoon of what it looks like, and there's an actual experiment that was done to physically show that this enzyme does rotate. So we know that it is actually rotating to generate ATP. And these are actually the microscopic images that were formed to show the rotation of this enzyme. They bound this molecule called everidin with a biotin to the top portion of this enzyme. That's actually what you're seeing in the image because they watched it move around as the whole enzyme moved. So this ATP is then, of course, it's made inside the matrix of the mitochondria. You can see that's happening through ATP synthase, but then we utilize other transport enzymes within the membrane itself to, for example, transport out ATP. One important one is this adenine nucleotide translocase. It's an anti-porter, meaning that as one ATP is moving out, it has to move one ADP in. So it's kind of transferring one ATP for one ADP. Another important one for this is the this phosphate translocator. Oops, this is a symporter, phosphate translocase symporter. And you can see it actually is one way that we can potentially rebalance out protons if needed and use these protons as well in movement of ADP to ATP. So this is translocating a fourth proton per ATP that is made to then move out that ATP. It is a symporter because it also takes in a phosphate as well. That phosphate, of course, needs to be used. Now, the net production of ATP by glucose, like how much ATP you get out of a glucose, varies. That's why you end up having these numbers that are something like 2.5 ATP per NADH and 1.5 for FADH. And it's going to vary also based on the organism that you're looking at. So, for example, in prokaryotes, their organelles do not segregate machinery. So they don't have these enzymes in different areas. And all the electron carriers can then easily feed directly into the electron transport chain. In eukaryotic systems, the segregation is going to prevent NADH from the cytosol directly entering into the electron transport chain. So sometimes we'll utilize our NADH in other areas. And so by having the electron transport chain in the mitochondria, we have to actually move NADH there to then make energy from it. So you actually have segregated pools sometimes of NADH and NAD+. Two methods are used to feed electrons into the electron transport chain from NADH from the cytosol. So if you have NADH in the cytosol instead of in the mitochondria, you can get it in there via the malate aspartate shuttle or through a glycerol 3-phosphate shuttle. And I'm going to talk about these two shuttles as ways to transfer NADH into the actual mitochondria where it can be utilized by the electron transport chain. Again, one of them we've already seen, which is the malate aspartate shuttle. This shuttle also is used to just move metabolites, right? It is taking aspartate and it is moving it from the matrix of the mitochondria. And then malate can move into the matrix of the mitochondria through a different transporter. Now, if you take aspartate out of the matrix, and you then are going to put it into the intermembrane side. If you transform it into malate and then move that malate back in, that actually uses an NADH. You can see here. Malate can then go back in, and malate can then be transformed back into aspartate, and that NADH can be released. So it's a way to get NADH or get electrons into the matrix from the cytosol without actually moving the NADH. So this is electrons being moved via aspartate and malate. And that NADH, these are two different NADHs that are being used and then made. But the idea is you ultimately are generating an NADH on the inside of the matrix via this whole shuttle. So it's an indirect way of moving NADH into the matrix. Another way is via the glycerol 3-phosphate shuttle. This is part of, if you remember, glycerol 3-phosphate is part of glycolysis. And via glycolysis, we're making NADH, right? And one way we can take that NADH is by transforming a dihydroxyacetone phosphate between a glycerol 3-phosphate. So these again are isomers of each other. We saw them in glycolysis. They can transform into each other. And through this process, we actually can move electrons through glycerol 3-phosphate into a coenzyme Q. So it's actually direct movement of electrons off of an NADH onto glycerol 3-phosphate, onto a coenzyme Q, and that feeds actually right into the electron transport chain. So this is a specialized way of dropping off electrons from an NADH that's not inside the matrix straight into the electron transport chain into complex 3. And note, if you do it this way, you don't go through complex 1, so you don't generate as much energy because you're not moving as many protons. This is what we mean by it's a variable amount of energy you could get. So this is again why we have this idea that the amount of energy you get, ATP you get from different pathways can be variable. For example, the NADH that you generate from glycolysis, the amount of ATP you could get from 2 NADHs is potentially only 3 ATP. That's if that NADH from the cytosol goes into the electron transport chain this specialized way into complex 3. Or that ATP can go all the way into the matrix. It can go into complex 1 and generate a normal amount of protons and ATP. So if your NADH is in the mitochondrial matrix already, you're going to just go straight to the electron transport chain and generate ATP. So things like the citric acid cycle, pyruvate oxidation, all these things are already occurring inside the matrix. But anything that's occurring in the cytosol and generates NADH, that, if you want to get then energy from that NADH, can potentially only enter the inner matrix and generate ATP through different means, giving you different amounts of ATP. And lastly, I want to focus on the actual regulation of this pathway. So we just spent a long time talking about all of how oxidative phosphorylation works, the actual electron transport chain, the actual chemiosmosis that occurs, and our final ATP that is made. Ultimately, this is regulated by substrate availability. Just if there is substrate, it will go. So if there is NADH and there is an ADP to then make ATP, this process will continue. Anything that is going to inhibit the actual process of ATP, so inhibiting the ATP synthase, particularly the F1 portion of it where it's making ATP, this will prevent hydrolysis of ATP, for example, during low oxygen. If you don't have that much oxygen, you actually can't finalize your transfer of protons through the electron transport chain, and thus you can't finalize making ATP. This is also only active at low pH. So that is a signal that there are a lot of protons on the side of the gradient where they need to be, right? They have made an acidic environment. If that pH changes, you actually will encounter stalling of electron transport. Now, an inhibition, if you do actually have inhibition of oxidative phosphorylation, you end up with an accumulation of NADH. And an accumulation of NADH, if you remember, will cause a whole feedback cascade all the way up to glycolysis, all the way up to phosphofructose kinase 1, right? Excess NADH inhibits phosphofructose kinase 1, which inhibits glycolysis. So anything that way downstream is going to inhibit oxidative phosphorylation will then signal all the way upstream to inhibit initial energy breakdown and stop it there. Okay, so here are some of those regulatory stopping points we've already talked about. You can see one's from glycolysis, one's from citric acid, and then ultimately oxidative phosphorylation. Again, the main signal here is going to be if you even have ADP around, you will make ATP. If you have too much ATP and not enough ADP, oxidative phosphorylation will discontinue. That's one of the main regulatory portions of it. Okay. To finalize this, I wanted to talk a little bit about the roles of mitochondria, because mitochondria itself is a very complex organelle. Again, we talked about how you can make energy in mitochondria, right? It's often called the powerhouse because this is the place where energy is produced. But we can use mitochondria and uncouple it from synthesizing ATP. Remember, electron transport chain will actually continue to go. But what will happen? There is another protein, this uncoupling protein, that can be utilized to use those electrons from the electron transport chain to instead generate heat. So instead of making energy, instead of making ATP, you then can use your electrons and use those electron carriers to generate heat. This happens a lot in babies. This is one of the ways that they will produce heat for themselves. Also, hibernating animals are going to utilize this to generate heat for themselves. Mitochondria also play a role in apoptosis. So, this is the process of programmed cell death. If you have any extreme damage, stress, reactive oxygen species, if there's some type of developmental signal that is processed by the mitochondria, you will then cause the mitochondria to rupture and release cytochrome C. Remember, cytochrome C is one of those electron transport carriers. It's part of the electron transport chain, but it has a different role here. If cytochrome C is found outside of the mitochondria, this is a huge signal to our body that there is some type of damage that is occurring. Cytochrome C should not be on the exterior portion in the cytosol of the cell. It should be inside the mitochondria. So when this release occurs, permeability occurs, cytochrome C can then be bound by these protease activating factors. So they're actually called APAFs, apoptosis protease activating factors. They'll bind the cytochrome C, and then through another process of caspases. These can then take what are called procaspases. So in their procaspase form, they do not signal cell death, but they are then cleaved into a caspase form through our apoptosis protease activating factors, which have now made a complex. And these ultimately will signal cell death. So it's a complicated signaling process, but the idea is that it starts off actually at the mitochondria and from the release of cytochrome C. Now, as I mentioned through this talk a couple times, mitochondria also has its own DNA. It has separate chromosomes and a circular mitochondrial DNA that is conserved in the mitochondria. This separate genome has 37 genes. It encodes ribosomal RNA, it encodes some transfer RNA, and it also is central to encoding enzymes that are part of metabolism. So here is where the actual encoding information for things, enzymes for part of the citric acid cycle and part of oxidative phosphorylation actually live. The mitochondria has its own ribosomes that then synthesize these proteins that are for metabolism. However, a vast majority of mitochondrial proteins are still encoded in the nuclear DNA. So not everything is in mitochondrial DNA, although a few very important proteins are encoded in this DNA. Mitochondrial DNA is maternally inherited, which is very interesting and unique. So the ability, your metabolism and your mitochondrial DNA is thus only inherited via your mother. And so any of these encoded proteins are going to be coming only from your maternal DNA. Mitochondrial DNA, again, is its own separate chromosome and it's actually circular. So this is what it looks like in its circular form. And you can see in each of these portions where different information is encoded. So information for complex 1, 2, and, or excuse me, 1, 3, and 4 are all on mitochondrial DNA. The actual ATP synthase enzyme is encoded here. And you can see also transfer RNA, ribosomal RNA, and kind of a control region for the DNA. This is just a general map of where things are. Now, what happens if you have mutations in your mitochondrial genome? It's a very small genome, and so accumulation in mutations here can happen quite quickly. Reactive oxygen species resulting from the electron transport chain can actually cause mutations onto your mitochondrial. Some cells will actually have more mutations than others. And because mitochondrial DNA is all separate to each mitochondria in each cell, that is why certain cells with their own mitochondria might have different mutations in them. So when cells divide, the daughter cells will have the same mutations as the parent cells. And sometimes these can eventually lead to a buildup of problems. If you have enough cells that divide often, like epithelial cells, skin cells, you have potentially a higher buildup of mitochondrial mutations. Other cells, like myocytes, which basically are stagnant until adulthood, in these cells, you don't really have this buildup of mutations that is occurring from constant division. Now, these mutations can lead to disorders. And again, that's more likely to occur in cells where you have more division. If you have mitochondrial genome mutations in things like uh, your um, your pancreas, for example, you can have diabetes produced. Things in your muscles will, of course, cause muscle weakness. You actually will not be as able to produce energy in your muscles. You can have atrophy. You can have nerve damage if it's happening in your nerves, liver failure, anemia. There's lots of different disorders. Again, these are separately connected to different types of mutations. Now, one form, as I just mentioned, in the pancreas is if you have mitochondrial mutations, can result in a very rare type of diabetes. Now, this is, I want to point out, very rare that you have this mitochondrial mutation. It is not the way that most diabetes is occurring. But ultimately, it's going to cause defects in oxidative phosphorylation, resulting in low ATP in the cell, and then insulin cannot be released from the cell. So, this is going to occur in your pancreatic cells. If you have this mitochondrial mutation in your pancreas, it can cause problems with insulin release. Okay, well, that is it for all of chapter 19. Now, that was a long lecture. This is everything on electron transport chain, how electrons are passed through the electron transport chain in mitochondria, the stepwise, what's happening in each complex, and how many protons are being pumped as a result of that transfer of electrons, how those protons then, through chemiosmosis, are providing power to drive ATP synthesis. And then we talked a little bit about the importance of mitochondrial DNA and how it is different and can lead to unique problems outside of our nuclear DNA.